Namaste everyone. This lecture is on transport across cell membrane. The human body is made up of 100 trillion cells. The cells are dependent on extracellular fluid, oxygen, nutrients for their survival. For cell survival, the cell need oxygen, nutrients from the ECF. The cell picks up oxygen and nutrients from the ECF. Within the cell, the metabolic activities happen where oxygen binds with the nutrients to form carbon dioxide and water with the production of ATP generation and heat generation. So this is the metabolic activity that is happening within the cell, and the cell for its survival need oxygen and nutrients, and these oxygen and nutrients are more in extracellular fluid, and their levels within the extracellular fluid has to be maintained, that is homeostasis. So now how these substances, oxygen and nutrients, will enter the cell, by what mechanisms, we will discuss now. And also, once the metabolic activity of the cell is over, the carbon dioxide that is produced as an end product of metabolism has to move out of the cell. What is the mechanism by which the carbon dioxide moves out of the cell? We'll explain. So, transport of cell membrane will happen by various mechanisms. So, first mechanism is passive transport. Passive transport. So, in this passive transport, there is no ATP utilization. So, this is like along the concentration gradient, the substances are moving from region of higher concentration to the region of lower concentration, downhill movement. So consider there is more concentration of substance here. So along the concentration gradient, these substances will diffuse, spread out. So movement of substances along the concentration gradient is passive transport. So there is no ATP utilization here. The movement is by only the kinetic energy. Kinetic energy. So the substances or molecules are in random motion. The molecules are in random motion. Consider these are all molecules and they are in random motion, and they when they come in contact with the molecule which is in static, some energy is transferred to this molecule and this molecule will set in motion. So this, because of the random molecular movement of these molecules, they spread out from the region of higher concentration. They spread out. So this is example of passive transport. So this is diffusion. So diffusion is an example of passive transport wherein molecules are transported across the cell membrane from the region of higher concentration to the region of lower concentration. So example for diffusion here is diffusion of gases. So now this is a cell. How oxygen will enter the cell or how carbon dioxide will move out of the cell is by simple diffusion. Simple diffusion. That means these oxygen or carbon dioxide are lipid soluble substances. They are lipid soluble substances. They cross the lipid bilayer. This is the cell membrane, lipid bilayer. They cross the lipid bilayer because of its lipid solubility. These gases are lipid soluble substances, and along the partial pressure gradient, they travel across the cell membrane. So for example, if the oxygen has to travel, the concentration or the partial pressures of oxygen in the ECF is more compared to that of intracellular fluid. So along the partial pressure gradient, oxygen will diffuse into the cell, and carbon dioxide, as an end product of metabolism, the carbon dioxide levels are more within the cell, and this will diffuse out of the cell along its concentration gradient through the lipid bilayer. So along the concentration gradient, this is happening without ATP utilization, and this is an example of simple diffusion. Diffusion of gases is example of simple diffusion. The other examples for simple diffusions are diffusion of ions across the cell membrane. So consider this is the cell. The sodium concentration is more outside the cell when compared to inside. So how this sodium will enter the cell, or potassium concentration is more inside the cell, how potassium will move out of the cell. So these are water soluble substances. So this cell membrane is lipid bilayer. Because they are water soluble substances, they cannot cross through the lipid bilayer. So there needs the integral proteins with pores. These are the integral protein, and there should be some pores through which the substances can enter or exit. So we'll draw a diagram of an integral protein. So consider this is a ion channel. So through the pores of the ion channels, sodium can enter the cell. So this is sodium ion channel. Similarly, there are ion channels. The integral proteins are acting like channels for potassium efflux along the concentration gradient. So the movement of ions across the cell membrane that is happening is through the utilization of integral proteins or transmembrane protein, which are acting like channels, channel proteins. Channel proteins. So this is also an example of simple diffusion, where the movement of ions along the concentration gradient happening through the pores of the channel proteins. Now let's talk about what is facilitated diffusion. Facilitated diffusion. So it is a type of diffusion where the substances are moving along the concentration gradient. The substances are moving along the concentration gradient, but they are facilitated by a transmembrane protein. Transmembrane protein will act as a vehicle or a carrier that will carry the substance into or out of the cell. For example, the glucose, how it enters the cell. Glucose is a larger molecule. This cannot cross the lipid bilayer on itself, or it cannot pass through the pores of the ion channels. No. So for this, the glucose entry, there should be a carrier protein with a binding site. So to the binding site, the glucose will bind. The glucose molecule will bind to the binding site of the carrier protein, and consider this carrier protein is called glucose transporter, GLUT, glucose transporter carrier protein. So once the glucose substance bind to the carrier protein, there occurs some conformation changes in this protein. So because of the conformation changes in protein, what happens? The outside portion will turn towards inside. The outside portion will turn towards inside, bringing the glucose to the inside. So glucose binding to the binding site of the carrier protein, conformation changes that is happening within the carrier protein brings the glucose to the interior of the cell. So this is how glucose from exterior will enter the cell, through binding to a carrier protein. So the diffusion of glucose along the concentration gradient is mediated or facilitated by the carriers here. The carriers have role in transport of substances. Similarly, other substances like larger molecules like amino acids are also entering the cell by the same mechanism, facilitated diffusion by the help of carrier proteins. Now, how facilitated diffusion is different from that of simple diffusion, let's see. What are the difference between simple diffusion and facilitated diffusion? So both are, see, this is the simple diffusion, and here we'll put facilitated diffusion. So facilitated diffusion is facilitated by carrier proteins, and examples for facilitated diffusion is glucose transport or amino acid transport substances binding to carrier protein. And simple diffusion will not, there won't be having any role of carrier protein. Carrier proteins are having no role here. The simple diffusion is across the lipid bilayer, transport of oxygen or carbon dioxide or nitrogen, and also through the pores of the ion channel pores, through the pores of the ion channel, movement of substances happening through the pores of the ion channels, like sodium or potassium or chloride. All these ions are entering through the pores of the ion channels are example for simple diffusion. So in facilitated diffusion, the carrier proteins are facilitating the diffusion process. In simple diffusion, there is no carrier involvement. In both simple diffusion and facilitated diffusion, whether there is a utilization of ATP energy is utilized? No. Why? Diffusion is a passive transport mechanism. Whether it is simple diffusion or facilitated diffusion, ATPs are not utilized. Now coming to what happens if there is increase in the concentration gradient or rate of diffusion, whether there is any affection. So consider now here, if you increase the concentration of oxygen, more and more oxygen here, all the oxygen can pass through the lipid bilayer and the rate of diffusion will not be affected. So consider this is a graph, and here concentration, increase in the concentration, what happens to the rate of diffusion? So rate of diffusion will not be affected. When you increase the concentration gradient, the rate of diffusion also keep on increasing linear fashion in terms of simple diffusion. But in case of facilitated diffusion, what is happening? If you increase the concentration of glucose on one side, whether the rate of diffusion will be proportionately more and more? No. This is because of limited number of carrier proteins and the binding sites. So rate of diffusion will be affected, and at a particular point, what is happening? Though there is increase in the concentration, the amount of glucose entering will reach a maximum value. The absorption maximum or Vmax is reached in terms of facilitated diffusion. So this is because of saturation of what? Saturation of carrier protein. So because of the saturation of carrier proteins, there is a Vmax and plateau phase in rate of diffusion. So how can you plot this graph for carrier protein mediated facilitated diffusion? With the concentration gradient, initially the rate of diffusion increases, and at a particular point with more and more concentration gradient, there is a maximum rate the maximum rate achieved, and this becomes a plateau in case of facilitated diffusion. So this is V maximum, or the maximum limit reached. That is the saturation, the property of facilitated diffusion. Now one more difference between so here in facilitated diffusion, carrier mediated process, second is saturation and Vmax, and the next property or the next difference here is facilitated diffusion will have competition. What is this? We'll explain. Competition in terms of facilitated diffusion. Let's explain this now. See, consider the facilitated diffusion is happening through the facilitated diffusion is happening through what? Through the carrier proteins. So this is a cell membrane, and this is the carrier, and this is the binding site for the carrier. Now consider this substance has to this substance has to, this carrier has to transport a substance A. There are few substances A. So this is a substance A, and if at all one more substance with the same structural is there, now this increased concentration of substance B will affect the transport of substance A. So consider these are all A, these are all A. So transport of substance A will be affected because of increase in concentration of substance B, and these will compete. These substance B will compete for the same binding site. Substance B will compete for the same binding site, and substance B will bind more, and this substance B will be transported more into the cell. So this is competition for the binding sites of the carrier that happens in case of facilitated diffusion, and the transport of substance A is inhibited by the increased concentration of one more substance B. So this is competitive inhibition of transport of A substance by increased concentration of substance B. Substance B will compete for the binding site and they will be transported more. So this is all about simple diffusion and facilitated diffusion. So these two are the types of diffusions and examples for passive transport mechanism. In the next lecture, we will discuss what is osmosis.